Great. So first, thanks very much to the organizer for inviting Professor Romero to this talk, and I truly apologize for him not being able to attend. So on his behalf, me, the typical PhD student, I'll come and do the job for him. So, and the reason is because I've been working with Nacho for the last 3 years and a half. I joined his lab as a master's student, stayed for my PhD, and I've been working with microRNAs in the concept of blood-brain barrier. Yeah, that one. So I've noticed that the talks today are that have been done are more focused on the role of neurons and how they're related to pathology. So we're gonna move a bit far from that topic and go a bit to vascular biology in the blood-brain barrier. So hopefully you're all related to the blood-brain barrier. But what is important to say about it is that blood-brain barrier dysfunction is a pathological feature common to many neurological disorders. And some of them are listed over here, and these ones are the ones that constitute the main focus of the lab that I work on. And in these pathologies, there is a trigger that can be common to many of these neurological disorders, such as cytokines and chemokines in, for instance, multiple sclerosis, but also another triggers, such as beta amyloid, that are specific to a pathology, in this case, beta amyloid, in Alzheimer's disease, that can trigger and then can initiate blood-brain barrier dysfunction that it is then characterized by an increase of paracellular permeability due to changes in the time junction organization of the endothelial cells, and also by activation of these endothelial cells that leads to actively extravasation of the leukocytes from the systemic circulation to the brain parenchyma. And these two events are the main characteristics of blood-brain barrier disruption, and these are the main focus of our lab. But for the purpose of this talk, I will be focusing especially in more detail on the molecular mechanisms underlying blood-brain barrier breakdown. And this is important because blood-brain barrier breakdown is one of the early events that happen in many neurological disorders. And it is not surprising that nowadays the blood-brain barrier is becoming more of a therapeutic target in order to try to tackle these disorders, and therefore it is important for us to understand what are the molecular mechanisms that underlie these CNS endothelial activations in neuroinflammation, or in our case, we're more focusing in multiple sclerosis. So multiple sclerosis is a chronic, typically progressive neurodegenerative disease that affects more than 2 million people worldwide, and is characterized by a loss of myelin and axonal injury. And in the earliest stage of the disease, there is immune cells trafficking into the brain parenchyma that occurs mainly in the meningeal blood vessels by crossing both the blood-brain barrier or the subarachnoid space, but also at the choroplexus by crossing the blood cerebrospinal fluid barrier. The presence of immune cells in the perivascular space can ultimately enter into the brain parenchyma, and these immune cells, together with activated CNS microglia and astrocytes, can mediate the demyelination and axonal injury that is typical of multiple sclerosis. In the later stage of the disease, the immune trafficking of the immune cells waves, but the inflammation condition remains inside the CNS and promotes this neurodegeneration. So I think we can all agree that this is tricky. The a loss of soluble barrier and enhanced transendothelial leukocyte migration are key hallmarks of multiple sclerosis pathophysiology. And together with this infiltration of leukocyte adhesion, we can observe that inflammation is exacerbated by the loss of integrity in the blood-brain barrier, and we can observe that in patients with multiple sclerosis, after injection with gadolinium, we can observe in those areas where there is an acute inflammation, there is also a leakier barrier that is it is shown by a brightness contrast of the gadolinium being transferred through the blood-brain barrier, as this chemical is normally not transported in the BBB, and this correlates nicely with the changes in the staining of attenuation CO1, that is a continuous staining in healthy blood vessels, but then the appearance of gaps in the staining appears in those blood vessels that have been stained from human brain sections of MS donors. And in this last graph, just to show you that the integrity of the endothelial cells is intact. This is actually the staining of the CO1 that is altered, that correlates with loss of BBB integrity. And this loss of integrity, it has been also been characterized in 2007 using different knockout mice for different genes involved in inflammation, and they found there is a positive correlation, regardless the genetic background of these mice, to increase, so they have positive correlation of increased blood-brain barrier permeability with increase of the clinical severity or score of these animals, as they've been induced with the multiple sclerosis model of multiple sclerosis, which is the EAE, or also named experimental allergic encephalomyelitis. So, given all this introduction, then we ask ourselves, what are the changes in cerebral endothelial cells in inflammation that might be in charge of these phenotypic effects that we are looking at? And for that, a previous PhD student before than me, they did a microarray study using a blood-brain barrier in vitro model, which is the human brain endothelial cells, or which now I'm gonna refer as a D3 cells, and challenge them to a pro-inflammatory challenge using TNF-alpha and interferon gamma. And they found that there were around 11,000 genes expressed in basal conditions in these cells, from which around 242 genes were downregulated higher than twofold, and 464 genes were upregulated. And so after an inflammatory stimulus, gene expression changes in the brain endothelial cells. But what can be driven these changes of gene expression? And we all are familiar with canonical processes of gene regulation, which are, in this case, transcription factors, and there is strong evidence lately that also low non-coding RNAs can modulate gene expression at the DNA level. Also, there are epigenetic modifications such as DNA methylation or chromatin remodeling that can silencing genes. But in these talks, I'm going to be focusing on a special type of regulation, which is at the post-transcriptional level, that involves what it is called microRNAs. And as mentioned before, lately evidence are suggesting that low non-coding RNAs could do this job as well. But let me take you through microRNAs. What are actually microRNAs? These little guys are short non-coding RNAs of around 20-25 nucleotides that are first transcribed in the nucleus as a form of a microRNA cluster if there are more than one microRNA gene together, or otherwise just one of these structures, and then is fairly processed by the DROSA complex to give to what is now called the precursor of the microRNA, with a stem loop structure and a complementary strand that then can be exported from the nucleus to the cytoplasm and further being digested by the dicer process to eliminate the stem loop structure, giving birth to two different single-stranded strands that can then be loaded in the RNA-inducing silencing complex and mediate gene regulation by targeting a specific region in the 3 untranslated region of the targeted mRNA and either degrade the mRNA or inhibit its translation itself. So, understanding this, we can agree that microRNAs can play a role in gene transcription at different time points of the canonical gene transcription pathways by either, for instance, downregulating mRNAs that encode for transcription factors, or mRNAs that encode for proteins that are related to RNA regulations, or in a more straightforward, basically degrading or inhibiting the translation of the targeted mRNA. So, so if these microRNAs can regulate gene expression, is there a link with multiple sclerosis? And this published this article came out in 2014, and we're coming back. Sorry. And so there was like a clear upregulation of different microRNAs in comparing different like brain, blood, and plasma with white cells, and they found that some microRNAs were shared between different tissue, and they found, for example, one of the common upregulated microRNAs was miR-155. So clearly, there is a deregulation of microRNAs in MS pathophysiology, and then we wonder, like, if whether do cytokines induce changes in microRNA levels in brain endothelium, and if so, are these microRNAs that we can select involved in modulating blood-brain barrier function? So, in order to do so, we go back to our blood-brain barrier in vitro model using the DC cells and stimulated with TNF-alpha interferon gamma, and we can see that if we do a time course experiment and using microarray data for microRNA analysis, we can see there is a time-dependent deregulation of microRNAs. If we treat the cell with a different combination of pro-inflammatory cytokines, then we come up with a short list of 3 microRNAs that are commonly upregulated, and you remember from the previous slide miR-155, but also we have miR-146A and B. And then from the 14 downregulated microRNAs, I would like you to draw your attention to miR-125B and 126. And also to point out that actually microRNA 155 was the microRNA that upregulated the soonest or the earliest in compared to the others. So I think at the time it was logical to start investigating its relevance in the pathophysiology of MS. And for that, we're using laser capture microdissection, in which you can specifically select microvessel from your brain tissue and isolate the RNA from it, and do then RT-qPCR assays. We analyzed that in those microvessels isolated from acute lesions in MS tissue, there was a significant increase of the expression of microRNA 155 when compared to microvessels isolated in regions with normal appearance white matter of both MS and healthy donor tissue. And this correlates with the increase of expression of microRNA 155 in the acute phases of the mouse model of MS, the EAE, as I mentioned before. So this suggests that miR-155 goes up when with MS, but as you might be aware, the blood-brain barrier is not only endothelial cells; they also have pericytes, they have the end feet of the astrocytes. We wanted to make sure that this increase of expression was specific of endothelial cells, and then by doing in situ hybridization, you can see that both microRNA 155 and PKAN1, which is a specific endothelial market, do colocalize quite nicely. So this suggests that miR-155 increase is probably in the endothelial cells, and then we go back to our question whether this miR-155 is modulating blood-brain barrier function. And in order to do so, we come back to our in vitro model, the DC cells, and then we can artificially modulate the levels of microRNA 155 by using a transfection reagents and transfecting precursors of the microRNA for 155 to increase the levels endogenously. And then we can see that after this transfection, there is an increase of paracellular permeability to paracellular tracers such as 50 dextran or 70 kilodaltons, which is higher of the scramble control transfected cells, but not as high as the cytokine-induced effect. And this functional effect correlates with the organization of time junction CO1 after overexpression of miR-155, which is similarly to the deorganization that occurs after inflammation by pro-inflammatory cytokines TNF-alpha and interferon-gamma. But only to trying to make sure this effect was real, we thought it was important to approach the experiment on the other way around. What happens if we block the levels of MIR155? And to do so, we transfect the cells with anti-oligonucleotide antisense of MIR155 and challenge the cells to TNF-alpha interferon-gamma. We can see that by blocking MIR155, we rescue partially the cytokine-induced effect in paracellular permeability and also the distribution of the CO2, so there is less gaps in the staining. So we know, at least in vitro, MIR155 is increasing paracellular permeability. So that takes us to think, okay, so if we knock down MIR155 in vivo, should we have a tighter barrier that might help to decrease the onset of the pathology? And we use a general knockout of a general knockout mouse and induce them with EAE. And we can see that the clinical scoring which is evaluated is significantly lower than the wild type, and that nicely correlates with a decreased permeability for fluorescence dyes in the EAE model, but also in a general inflammation model, which is injecting LPS. And this is just to show you that only in the wild-type mice with EAE there is an increase of expression of MIR155, and our knockouts were actually knockdowns. So we know MIR155 can modulate BBB, but I think what is interesting, especially from the therapeutic point of view, is trying to understand what targets can be modulated by MIR155 that are actually giving this phenotype. And to do so, using transcriptome analysis, we identify some of the pathways that were deregulated after overexpression of MIR155 in the brain endothelial cells, and then we can see that some of the downregulated genes are related to focal adhesions, to time junctions, and therefore to globin barrier integrity. And after come up with a list of candidates, we're using bioinformatic prediction databases. We came out with 4 genes, which are DOG1, syntenin, annexin2, and claudin1, all of them focal adhesion, time junction proteins that we were able to validate that were actually targets of MIR155. And then if you knock them down in the brain endothelial cells, you also get an increase of paracellular permeability, which suggests that those are the genes that at least partially MIR155 is regulating. And in this immunocytochemistry, we saw for the first time the focal adhesion knockdown as DOG1 and syntenin1 actually changes the subcellular localization of time junction CO1, and that was the mechanism by which they were acting as modulators of BBB. And this is published data that is available in the Human Protein Atlas, and then you can see that all these targets are co-expressed in endothelial cells in this database, which suggests that they are actually relevant biologically speaking. And and that's the story of MIR155. So at that point, that was published. MIR155 modulates globin barrier permeability and through these targets. And also 2 years ago, they also published that, we also published that MIR155 overexpression also modulates leukocyte adhesion, but I won't get into that in detail. And at this point, and that's one part of my PhD, I'm working on what are the mechanisms that increase of adhesion is for. So if you remember my previous slide, I talked about the other few microRNAs, and we've been talking about MIR155, but what about the second highest upregulated microRNA, in this case, 146A and B? And Dongsen, that was a postdoc in the lab of Nacho, did a very wonderful work characterizing in the same way as I have explained with MIR155 the correlation of MIR146 in both in vitro and in MS and in EAE model, and you can see that its levels are increased, and that its levels are specifically in the endothelial cells by in situ hybridization. But what is interesting in this case is that if we block MIR146 instead of having a decreased paracellular permeability, we also actually had an increased paracellular permeability, which suggests that probably MIR146, instead of being a pro-inflammatory microRNA, is actually an anti-inflammatory microRNA. And a bit more of the investigation that was carried around this microRNA is regarding to its role in leukocyte adhesion. And Dongsen demonstrated that the overexpression of microRNA 146A decreased the adhesion of leukocyte cells to human brain endothelial cells, and this was due to because it was preventing the the NF-kappa beta transcriptive factor for happening, and this NF-kappa beta is one of the main transcriptive factors that regulates inflammation in any cellular type, and it was due by some known targets of NF-kappa beta suppressors, but also he identified novel targets, in this case NF85 and the RHO-A protein. And finally, if you remember, there were like a few down-regulating microRNAs that I point out in the previous slide, and in the lab they decided to collaborate with Amsterdam University, with Professor De Vrie's lab, and they culture cells with the pro-inflammatory cytokines, but also cells cultured with astrocytic conditioned media, which is believed to have anti-inflammatory properties, and then compare which microRNAs were downregulated by pro-inflammatory stimulus and upregulated by anti-inflammatory stimulus to identify possible housekeeping genes of blood-brain barrier function. And they came out with the ones I pointed out before, MIR1 to 5B, and microRNA 1 to 6, 3P and 5P. And this is believed to be housekeeping microRNAs that in a physiological role, they are maintaining the homeostasis of the blood-brain barrier, but after inflammatory stimulus, its levels go down, which allow inflammations to occur in the endothelial cells and promote both blood-brain barrier permeability and leukocyte adhesion. And just to sum up, we have a pro-inflammatory stimulus, in this case TNF-alpha interferon gamma, that leads to a regulation of around 10 microRNAs in the highest top list, and a regulation of more than 100. And I've been talking about MIR155, 146A, the housekeeping genes, MIR1 to 5 and 1 to 6. And all of them can regulate different points of gene expression that ultimately will lead to increase of paracellular permeability, but also leukocyte adhesion and modulating the blood-brain barrier function itself. So these are just the hallmarks of today's talk. So I think we can agree today there are some microRNAs that we can call inflammeres, and their expression gets either up or down, and expression gets upregulated after inflammation. But we also have some microRNAs that are considered to be housekeeping genes, which expression goes down after a stressful stimulus such as inflammation, and therefore increase the chances of the cell suffering for the inflammatory stimulus. But I think it's important at this point of our research to say that, but if microRNAs are to be considered for targeted of prophylaxis and therapies for blood-brain barrier dysfunction, we need to know first the combinational effects of targeting all these microRNAs in specific CNS pathologies, and also what will be the off-target effects, as they can modulate many different targets. And I just wanted to give you a bit of a brief what we are doing right now in the lab. So obviously we're still investigating the molecular mechanisms of neuroinflammation mediated by microRNAs, but also we are moving to look into how extracellular vesicles, and in this case exosomes, can modulate blood-brain barrier function in inflammation, and also we're doing some analysis of microRNA and low non-coding RNAs levels in the blood-brain barrier in healthy aging. And we are a very welcome lab, so if any master's student would like to come and do an internship, you can come and enjoy sunny England. And so that's everything from us. I'd like to thank all the collaborators that NATO has had over the years, especially Professor De Vries in Amsterdam, David Baker in Queen's Mary of London for inducing the AE in our mice, Basil Sarak, Professor Sarak in Sheffield University for supporting and funding our research, obviously everyone at the Open University, and most of the work in May 155 has been done for Dr. Lopez and Camila Ceruti and Dr. Wu. And thanks everyone for listening, and I hope you have enjoyed a bit this different talk of neuroscience. And if you have any question, I'll be happy to answer.